Okay. Oh, hey, look at this. Huh. Already we got the vaginicola out. Wow. Let's see. I need to make sure that my camera settings are all still okay. I think they're good. I think we've got everything set up normally. There we go. I took my camera off my my microscope this week. I did a little bit of filmography for my work, which was fun. What's up, Teeny? How you doing? Hey, Rajiv. What's up? Good to see you in the chat. Let me see. Let's pop out the chat so I can see that. Okay, get going. Give me 1 second. Okay. Yeah, here we go. So, today's starting sample is critters from basically there's a mud puddle. There's a park called like Baylands Nature Preserve or something like that, and there's kind of just a puddle near the trail that I guess like fills up at high tide or something like that, and it's always got a nice like thick mat of this algae, this green stuff here, growing on top of it. And then at the bottom, the sediment is just this like horrifically smelling black sludge. And but it seems to be absolutely packed with life. Lots of cool marine critters. This, I think it's vaginicola. If it's not, it's very similar. I think to identify them, like you have to look at like kind of the shape, the opening of this like vase, and then how it comes down at the end, how it grows out. These are cool. Cool little ciliates. You can see if you look real close, you can see some striations, some lateral striations. Hey Amy, welcome in. Teeny's asking about other Discord communities for science. Let's see. Well, I only really know like a couple Discord communities for that like other streamers have. There's some other like science streamers. There's one that Pacific Plankton is associated with called the Knowledge Fellowship, which I can link to, although I think it's kind of inactive. At least I don't ever see much happening there. I'm sure there's other science ones out there. You just gotta. This is the really my kind of main gripe with Discord is that it's difficult to actually like find stuff. There's a microscopy Discord, like a general microscopy Discord. If you guys didn't know, I'm on that, and like I only found out about that like when someone casually mentioned it to me on Reddit. So that's it's a little unfortunate. Whoa, check out what the cilia are doing on this little like Whoa, check that out. I think they must be like spinning in sync with So I'm recording this at 60 frames per second. So these must be like spinning in sync with that. I wonder, can I change? I think I can change the shutter speed. Let's see if I go like really fast with it. Does it? I guess it won't matter. I need to change the actual frame rate. I can't do that easily. Let's see. So that is right underneath the cover slip. So we can go take a look at that with a nice high magnification objective. We will use our 40x for that. And let's go over to the 40x. Turn that down. Yeah. And there's this interesting little critter. Let's see, I want to go this way with it, I think. There we go. Check this thing out. It is, you know, I don't think it's actually a halteria. I think it's got too many little feeler things. That's right, Teeny, the Knowledge Fellowship. A weird little critter. Okay. Oh, it's kind of like, yeah, it's kind of migrating over this. It makes it more difficult to get imaging when it's got something right beneath it. Get good imaging on it. Oh, we can look at some of this other stuff here, some of this plant matter. Ooh, there's a nice, is that an epithemioid diatom there? It looks like it. It's got kind of the bumpy surface. What's up, Shahab? Welcome in. We got a coleps here, and this coleps, we can tell, has recently split. You can see it's sort of like only half dressed. So on the left side of the coleps, it's got like its protective calcite armor, and then on the right side, it's kind of unprotected right now. Ooh. Hey, here's a nice Dinoflagellate. And on this Dinoflagellate, we can see the transverse flagella. Ooh, it just started going real fast. Let me bring my go here. Make sure my curlers might be a little sketchy today. It's a pretty thick sample. But there we go. Yeah, check this thing out. So yeah, dinoflagellates, it seems like they typically, or at least they tend to, they'll have like one loose flagella that they kind of use as like a propulsion device. And then they have this transverse flagella that like coils around them. And I know some of these can be harmful algal bloom critters. So that's cool to find. There's a cool ciliate. All right, let's see. How many dust spots can I count? I count 1, 2, 3, 4, 5, 6 dust spots. We should figure out like how many dust spots before I actually have to clean my camera sensor. How many is okay? It's not something I particularly enjoy doing. Usually kind of finicky and difficult. Here we are back at the 10x objective. Ooh, look at that ciliate. That's like a... It looks like a Loxophyllum type of ciliate. Go to the 20. And one of the challenges I think with this sample is that like there's so much stuff in it. Oh yeah, I think it's pretty deep. Oh, there's another one up here. Okay, actually, yeah, it's just a little. It's looking a little soft just because of how deep the slide is, or the sample is. Okay, we'll go back to the 10x. Yeah, there's a lot of water here for me to focus through. Could have probably made this a little less thick. But I wanted to get, one of the things I wanted to get on this slide that I didn't have the last time I was doing the brackish water was some of the algae. Well, there's some oscillatoria, some cyanobacteria. There's another one of those dinoflagellates. And another, interesting, got a fair amount of these. I'll have to vet these with Pacific plankton. There's an interesting little critter. An interesting little ciliate. It's got a very distinct vacuole there. Yeah, look at this thing. Where are you? Oh, it's coming up towards the surface. Oh, it's kind of spiraling up. Whoa, look at it. It's just sort of suspended there. Is it like stuck on something? I don't know. Wow, that vacuole is just very spherical, very like stand out. This is one that's like, okay, I could probably get an identification on this. Or maybe there's tons of them that look very similar. But at least to a genus. Nice. Oh, there's our sulfur fixing bacteria there. I think that's probably vaginicola or something similar. This colorful strand here. Yeah, I think these diatoms here are like epithemioid diatoms. They have that interesting like little kink to them. Is that an amoeba there, perhaps? Yes. We got a little amoeba. Hi, Jeremy. Welcome in. Oh, this is well, that's a nice little diatom frustule there. This thing. All right, let's go back out. Let's go back to the 10x. Got our exposure back down. DIC prism into the right spot. Open that up. Hey, we got a nice Euplotes here. So one of the interesting things is, so I have this this book about for identifying marine interstitial ciliates, which are ciliates that are marine, but also are like they live in the sand. Basically, they live in or at least in sediment, and whoa. And one of the interesting things to note is that like there are like pages of Euplotes, just different species of Euplotes, and they all, well, not all. Some of them are, I guess, kind of distinctive. I think there are some Euplotes that actually don't have cirri, which is interesting. But there are just so many species of these. Ooh, you're a chunky little crustacean. This is like a very chunky looking Nautilus or something. What are you up to, little buddy? What are you going to turn into? Right now, I'm using the 20x objective. And let's see. Why don't we try for a little bit of Darkfield? Let's see if we can get that working. Let's see here. There and boop. That might be. It's okay. It's just very, it's very deep for this objective. Maybe if I go to the 10x, yeah, that's a little bit better. It's like a cross between a Nautilus and a copepod, and a little extra chunky than either. And it's got kind of a, like, there's almost like a little, see, there's like a shell, and then there's kind of like a tail portion of it too. Interesting little critter. Oh, there's a nice, actually that cyano looks pretty cool. in the Darkfield. Hey, the bobsled, welcome in. Thank you. I'm glad you're enjoying it. We do strive for gorgeous visuals here. There's our Euplotes again, and our little duder. Look at all these coleps that are just kind of sitting on the on this like seaweed or sea algae. I don't know. I don't know what the preferred nomenclature is. But you can get a nice view of their. I'm getting just a little bit. It's just because it's such a thick sample. I can't really get my Darkfield tuned properly. It's okay. We zoom in a little bit. Yeah, and you can see these coleps are just kind of. They seem to be kind of docked with this this algae here. Algae is more correct. Huh? Okay. What about kelp? Shahab. What's kelp? Look at these guys. I wonder if they're like consuming it or what. And then we got some little diatoms as well that are sort of stuck onto this. Ooh. And here's a hypotrick, which is standing very still, which is unusual for hypotricks. Look at that. You can see it's got like 2 rows of cilia there. That's probably important for identification. And it's got kind of like an oral groove there. You see that beating away. Oh, here comes Frontonia. Frontonia lucas, I believe, or something similar. Oh, what's up with my Darkfield? It's kind of wonky. Oh, it's just because I think it's too much. Go to the 10x again. There we go. Yeah, wow. Yeah, look at that Frontonia just like climbing up that. Kelp is one of the largest algae. One of? It's not the largest? What's the largest algae? Can you eat kelp? I think I've heard that you can like eat it. I'm not just talking about like normal seaweed. My little cyanobacteria friend here. It's a very large bacterium. These are cool too. These will, if you hit them with like blue light, strong blue light, they'll fluoresce red, which is something I've been meaning to do. I've been meaning to get my fluorescence microscope set up. I've had been working on some other projects. Well, the next time I'm at the beach, I won't pack my lunch. I'll just grab some kelp. Yo, hey, look at this. Whoa. So we have, I believe, some structural color happening right now. It's too deep. It's too deep. Let's see. Let's see what happens if I go back to DIC. Maybe I can kind of carve this thing up a little bit. It's one of the advantages of DIC is that you can kind of slice things better. Wow. So I'm pretty sure this is a diatom frustule. But I can't see the poroids on it. Oh yeah, I can see there's like a perimeter of little spiny things, it looks like. So I'm assuming that this has some pore structures that are just really tiny. And I think at this depth, I really can't, like, I can't actually image them with like a higher numerical aperture. So it's interesting. Let's do a little bit of Brightfield. We like a little bit of Brightfield. Go here. We'll use a ND filter to kick the field down a little bit. Bring this in and get our curler back. There we go. Give us a nice even illumination profile. Let's see. And we'll go down to like right there, about. And bump that up a little bit. There we go. Brightfield, always nice for kind of seeing the true colors of things. Hey, there's our Euplotes friend. Correct, Shahab, that was DIC. I have not done phase pretty cool in phase contrast, so might check that out. See, we're on the 10x objective right now. Just taking a look around. Seeing what we see. Yeah, a good amount of these Dinoflagellates. And then these big Frontonia here are so cool. Let's see if we can come up. Well, we can clip a little bit. It's no problem. Oh, see ya, Teeny. Thanks for stopping in. Oh, there's that hypertrick from earlier, I think. And you can see this stuff here. So I guess I don't remember if I mentioned it. Like the sediment is black, like dark black, and it smells incredibly of like sulfur and just kind of like generally like burned matter almost like. And so here in Brightfield, you can really see like this stuff that's dark. I mean, it's just that's the stuff, and it's very black. I don't know what the composition is. There's something here. That's kind of an interesting little structure. There's some movement here. Suspicious movement. What is that? Anything in there? Is that like an amoeba there? I think there's something. Yeah, I think there's something kind of oozing through. Oh, hey, there it is. So it's interesting. So it looks like there is an amoeba that is like globbed onto a little strand of cyanobacteria. Oh, there it is. We're at the 20x objective now. I think it's not what's causing the move. Oh, maybe this whole strand of cyano here is moving. Maybe this thing's kind of tugging on stuff. Go to the 4x here. See what we see. There's a nice big oscillatoria. Yeah, look at all these little dinoflagellates buzzing around. I think that's what those are. There's a... Okay, that's that. Whoa. Oh, here's one of these. This is one of those like weird shrimp things with the big 2 front feet. Sort of like they're like little mantis-looking things. Do a little polarized light. A little polarized light microscopy. Seems like a good play. Sound play. Boom. Pop this bad boy in. There we go. Let's take that off. And bump this up. Oh, wow. Awesome. Let's see. We can do a little switcheroo. And let's see. Yeah, this is my thing here. And we could do a little bit of that. Let's see. Let's make sure we're at extinction here. Yeah, we just come in here. That's nice. It's a nice little vibe. Yeah, check that out. So this is super cool because you can see the musculature in these little legs. And you can see that it, with the wave plate in position, the orientation changes how the color that they illuminate in. brighter. A little brighter. This is with the 10x objective right now. I could bring this. I'll just bring that down a little bit. And then the shell appears to be pretty birefringent as well. Right there, it's very bright. Well, mostly like these little feets. These little feets here, and these like cool little muscles. This is the 20x now. That's right, E-Packer. So muscles, like yeah, muscle fibers tend to be pretty significantly birefringent, and so I'm using polarized light right now, and so they show up in the polarized light. And you can even, I think, if you like look closely, you can kind of see that they are like contracting and stuff. Kind of twitching a little bit. We'll do some more. Oops. Whoa. Go right there. Ooh. Hey. Kind of change up the mood a little bit. This is. There we go. Sorry, we've got at the 4x, we've always got a bit of a vignette. But this is still better for kind of. Oh, look, we have a snail. We have a little snail. And it has an extremely birefringent shell. Which, let's see, if I turn down the brightness a fair amount, then now we can actually see the shell, which I guess must be quite honestly, like probably really nicely crystallized, like, you know, it could be like calcium carbonate or something like that. I don't know what snail shells are made out of, but something birefringent. Okay, for this snail, since the polarized light is kind of blowing out the rest, I'm gonna go for Darkfield. So let's see, we'll just pull this out, and then let's open this up all the way. There we go. And then we'll just raise our condenser up until we kiss the slide. Bring it back down. Maybe that down a little bit. And we'll go to the 10x here. Oh, it's kind of, it pulled back up in its shell. You can see the eye right there. I wonder if I'm gonna add a little drop of water near the slide or near the cover glass, and we'll just see if that prompts any behavior from our little snail friend here. May not. But at least maybe this will keep it from getting super crushed. And it's cool, you can see on its shell, you can see that there are some diatoms anchored on that. There's kind of a little community of microbes on the shell. And there's our snail friend, just kind of hiding away in there. go to the thorax again. That's cool. You got a little snail. Snails are always fun, you know? I like a snail. Right there. Ooh, we got a Frantonia party here. That down a little bit. Sweet. Hey, Andy. Try not to crash your cart into anybody. Diatoms is not responsible for any accidents of incurred while watching while driving or something like that. Oh, this is cool. I like when I love this little optical effect here. Seeing these these Frantonia through the algae. Whoa, it's kind of trippy, huh? Anybody else seeing that? It's sort of like it almost breathes when the Frantonia move underneath. How about we go back into polarized light? Oh, why aren't you going in? There we go. Just curious to see how that looks. So let's put this in here. There we go. It's interesting, the Frantonia, they don't have a soup. They have a couple little crystals in there, a couple bright crystals, but they have more of this not really tremendously birefringent stuff. So they're not that bright overall. Coleps, they'll have some nice birefringence going on in their little armor shells. Let's see, again, we are at the 10x lens here. I'm double zoom, that's what's going on. Thought that was a little tight. I mean, there's one that's kind of bright. Wow, it's very pretty. All these little sparkly bits, these sparkly birefringent bits in the what is again, I must reiterate, horrifically stinky black muck. So it's cool that in polarized light it becomes pretty. There's a little rotifer. Frantonia. Ooh, hold on, that's just a thing. There's our snail friend again. Let's Still hiding up in there. Whoa. Oh, check this out. This is some little crustacean here. I should be able to at least identify these. These are like common enough, but it's gonna hide from me. But check out that eye is like super showing up in the polarization. Yeah. All right, stay in there. Fine. Go check in on our yeah, this guy again. Maybe we can give this one a little water as well. We can just really load it up. Give it a whole bunch of water. There we go. And just see if anything, if it moves a little bit more or what. I think it might be kind of squished though. It doesn't look super hot. Anything come in with this? I wonder. Yeah. I can try my. I can put in my my actual wave plate that came with this polarizer. I think it's got like a little smudge on it. That's fine. It's whatever. Let's see. Let's drop this down. This is was this way. Oops. I just put like a nice fingerprint on there too. Cool, cool, cool. There we go. So this is I forget. There's a name. There's like a very like technical name for this this offset here. This kind of magenta background. But it's interesting. It kind of the muscles don't seem to be as clearly polarized anymore. Some of them are, some of them aren't. Very colorful on the critter though, that's for sure. Like that. There we go. Go to the 10x. Yeah. Interesting. Is that like a little ciliate or something that's sort of stuck on its nose there? Interesting. Maybe it's eating it. Oh, there's maybe a little ciliate as well. And it's pretty common for these like crustaceans to have a lot of hitchhikers, a lot of ciliate hitchhikers. Cool. Go back out. Oh, hey. Where'd you come from? Snuck in. Little guy. Let's take the wave plate out. Whoa, yeah. You can still see the eye. Wow, check that out. So Shahab, the filter that I'm using is it's a wave plate, and it's I forget the technical term. It's a wave plate. It's like the official kind of insert that comes with the polarization attachment that I have, and it says 530 nanometers on it. And I believe the reason that you're seeing magenta light when I insert this wave plate is that I'm basically cutting out the middle wavelengths. So I'm cutting out the greens, basically the 530 nanometers. Those are being cut, but other wavelengths can get through, and so you end up with what's left over mixing, and so that's basically like the red side of the spectrum and the blue side. I think that's a not completely incorrect explanation of what's going on. Check this. Oh, there they are. Wow, they're like congregating over here now. These things also totally remind me of the little robot things from the Matrix, the ones that they like from the first movie, they stick inside of Neo's belly. Okay, I'm gonna prepare another slide now from a duck pond that was right next to the shore. It's like there's some kind of like water treatment thing going on with it. I'm not exactly sure, but it's like much less smelly. In fact, I was actually thinking that maybe there wouldn't be anything in it because it was just like it just seems very clean and clear, like maybe it's treated or something like that. But I took a peek at it already, and it seems to have plenty of stuff. So we're gonna try and get some of that out. So give me a couple minutes here. I'm doing what I kind of refer to as like a mini extraction, where I just take like a glob of like algae and debris and whatnot, and I put it on the slide, and I just I add some water on top of that, and then I just sort of squish the algae on the slide and whatnot. And then I take it off, and theoretically I'll have washed specimens out on there. In fact, I think a pretty good job with this one, so I'm going to try and take a risk here and use an actual number 1.5 cover slip. Okay, and we'll see if I made this thin enough to take advantage of the 1.5-ness of it. Oh, hello. Hello. We got critters. Oh, we got a lot of critters. Oh my god, nematode! Nematodes, always super bright in polarized light. I think there's an Arcella there. Ooh, another one. Okay, so we got lots of Arcella, it looks like. And we're at the 4X now. Wow. We got nematodes. Ooh, we got... What is that? Whoa. What's going on there? I can see some like red. So now we're at the 20X here. I'm not sure what that red stuff is. Oh, maybe those are, this could be structural color from the diatoms. I've seen diatoms show up red before. But maybe we should go to not polarized light to start with, and that'll give us kind of a better read on what exactly is going on here on this slide. There we go. Oh, we got some rotifers. Ooh, look! We got a little flatworm buddy. Take a look at our flatworm buddy. Let's bring our iris down like that. And we can pop this up a little bit. Oh, we should adjust our color since we've been changing a lot. There we go. And there we go. Lovely. Cool. Got kind of a look at the hind section of this, the posterior. It's got like some definite interesting kind of structure going on there. Look at that, yeah. It's got like little like nodules there. It looks like it's almost adapted to be sort of a like a sessile worm, you know? So we're like kind of like a stentor where it will just like anchor at one spot. I don't know if that's actually true. I'm not a biologist, but this thing's neat. Darkfield? Is that a play? Is that a play? Oh, we can take this off. We don't need that. Oh my goodness gracious. Oh my goodness gracious. It just pieced out. I wonder, so I just removed the neutral density filter. I wonder if I made it like too bright for this guy. Wow, this thing, this is a very strange little worm. I just don't normally see flatworms that have like this thing going on at the back. It really behaves a lot like a stentor. Look at that. This thing's super cool. What do we want to do? We want to do a little bit of, we can quickly go into polarization. So just pop that in like that. There we go. Maybe open that up. Oh, don't go anywhere. Hey, you get back here. Stop right there, criminal scum. Wow, look at this pretty boy here. You've been eating some sparkly stuff. Look at that bokeh. Coming out a little bit. Coming out of focus. Dreamy. Wow. Boom. Okay, I'm gonna just... Boom. Go like that. Zam. Boom. Hit it with the wave plate. Give it a little color. This hair, this like fiber here has got some polarization response as well. Wow. Whoa, check out in the polarized light here, like the membrane of this thing is interesting. Oh, it's on the move. On the move. Let's bring this down. We're at the 20x now. It's so pretty. Sure pretty. Oh, are you gonna move again? Oh, okay. Ooh. Let's see. Where do I have yeah, right there. Get this orange background going on a little bit. Maybe drop this down just a little bit there. Looking pretty neat. Pretty neat. Nematode. Hey, what's up, Mike? Welcome in. This thing is so cool. I love this worm. Big fan. Big fan of the worm that behaves like a stentor. So let's see, it's right under the cover slip. So let's see if I go change my 40x to that. Should be able to get some 40x imagery. There we go. Wow. Let's see if I should be able to open this up a little bit there. Drop that down just slightly. I think right there is fine. Yeah, sparklies are definitely one of the high tier attributes of polarized light microscopy. Get a little pink going on here. All right, pretty high ISO right now. That's all the way open. If I bring this down a little bit. Got some like interesting kind of vacancies here in the back. Can't tell if that's food it's eaten or what. I'm gonna give just a little extra water to the slide. I think this thing's maybe getting a little bit squished. So there we go. Yeah, see, look. I just put water on the slide and like look what happens. It all of a sudden gets like way narrower. Oh wow, look at you can see like the cilia on it. This thing's gonna start moving now, isn't it? It's like, oh, I can move. Okay, Not hanging around here anymore. Whoops. Let's go back to the 20x. Okay, how do we want to look at this thing further? We could do DIC. I'm kind of curious. Let's see. While we're here, I don't think there's anything preventing me from going into phase contrast. This is interesting. This is with the 10x phase contrast annulus. And then let's just take this out of here. Let's get that going. Let's turn our ISO way back down. Although we're going to have to turn it back up for phase contrast. So we just come through here. There we go. And yeah, then we just get this bad boy going. Let's open that up all the way. And here we are in phase contrast, which just kind of makes this thing look kind of muddled, generally speaking. Got some other stuff going on here. We got some Marcella. Got our nematode. Is this, okay, that's good. Which objectives were those, Mike? I don't recall exactly offhand. This is weird too. I don't understand why I'm at pretty low ISO. I usually have to go higher for phase contrast. So I'm like, what did I do? Maybe because I usually use phase contrast with the polarizer in, just for convenience's sake. Oh, you can see the hairs on this thing. Like when they stop moving there, you can see the hairs in phase contrast, the cilia. But let's go ahead and get back into DIC mode here. So we'll pop our polarizer in. We'll take our polarizer slider out and put our DIC analyzer in. Turn that down. Uh-oh, Mike. Sounds like somebody's got microscopy fever. Somebody's got the microscope fever. It's okay. You're in a safe place. Yep, there's our little worm friend, the rotifer. Let's see, Mike, if you're lagging, it looks like everything's good on my end. At least I haven't dropped any frames or anything like that. So I think it's on your end. Ooh, there's a nice heliozoan. And we got some more nematodes. We got a little ciliate there. Got these interesting rotifers here. These are ones that I don't often see. Ooh, there's a bit of a party over here. Bit of a microbial party. Why wasn't I invited? I think, okay, we got that. Come right there. And we just kick our ISO way up. Boom. Down a little bit. There we go. Yeah. Whoa. What is that? This looks Loxophyllum-ish. Look at those little spines on the perimeter of this thing. You guys notice that? So if I go right here, look at those. It's got like little spines that don't appear to be moving. That is surely something that could help identify this. Is this like a rotifer? No, it's not a rotifer. It's just a ciliate, right? Oh, is this a It could be a metapus, or it could just be twisted up. It does kind of look like it might be twisted up. Hard to say. There's a fun little flagellate there. Pretty motile little guy. Arcella. A lot of Arcella or at least just Arcella like testate amoeba shells. None of them seem to be moving too much. Yeah, what happened to me with BH2s, Mike, is like I just started realizing that like, oh, it's always like way cheaper. Like rather than going and buying like the parts that I need, you know, that have been like parted out by people who know what they've got, it's like, oh, I can just buy stuff, you know, from like surplus shops. You know, I can buy like whole microscopes that have like the individual components that I want. And that tends to like work really well. Ooh, hey there. Hold up. Is this oh, there's like an amoeba. I was thinking, is it like Vampirella? But no, that's a cyanobacteria. Check out this amoeba. Whoa. We got the 40x now. And check this thing out. I don't know what the story is on this little thing. It looks like just kind of like a little algae colony. This is right under the cover slip here. Yeah, Mike, you might you maybe can try dropping the quality down to like 1440p or something like that. Because it is, you know, a 4K stream is kind of a lot to it's a lot of data. Ooh. There's another weird looking ciliate. Oh, but it's got a cyrtos. That ring there. Wow. That's like an oral apparatus. Oh, there's another one. Ooh. I think these things are generally, like when you see that, they're generally diatom eaters. Ooh, here's a nice big nematode. Thanks for letting me know, ePacker. So Mike, while you're at it, buying new microscopes and all that, you might as well also upgrade your internet package or something like that, you know? And since you're already in the doghouse, you know, just go full blast. Yeah, here's one that's got a diatom inside of it. Oh, these things look real haggard, though. They're not the prettiest ciliates ever. But there are a lot of them. Oh, is this? Oh, that was like a side view. What the heck? Look at this thing. What kind of... This is like... It's like the Pontiac Aztec of ciliates. Just... Who designed you? And why? Ooh, look, it's one of these things. With the little oral apparatus deal. That's cool. Let's see, we're still on the 40X lens here. Hey, Amy, so just FYI, there is when I'm doing 4K streaming on YouTube, there's about a 30-second delay between comments, well, what's happening on my slide, and then comments. So I'm happy to answer questions, but if you can provide just a little context, it's helpful. Because I don't exactly know what it you're referring to. Look at these testate amoeba shells. Looks like, look, yeah, it's got an amoeba inside of it. It's alive. It's just kind of chilling. Oh, the cyrtos, I think, is what you're... Yeah, the ring, the little dots. So the cyrtos, I don't know exactly what... Ooh, there's some nice cyanobacteria kind of matted up with these diatoms. There's a whole, like, group of organisms that have a mouth apparatus like that. And I don't know what, you know, design advantages it has, but in general they use it to eat up diatoms. That's a nice view of the test there. Ooh, we got some purple sulfur bacteria here. That's what those are. And those, I've definitely seen, like, critters like to eat those. I'm going to switch back to a lower magnification here. Let's go back to 10x. And let's do some more exploring of the slide. Oop. And our little nematode friend. Well, it's maybe getting a little dry. Fix that, no problem. I think I measured the salinity of this water to be about 15 parts per thousand. Nematodes, I mean, they're the masters of spazzing out. Like, why though? Why do nematodes spaz? Does anybody have an answer for that? The purple sulfur bacteria is just like the little pink circles, the little pink balls. That's a nice little ciliate there. Wow. Another nice ciliate. Oh, look at this. So these guys, they really like the bubble here, it seems. I guess they're looking for some oxygen. I definitely am going to make a petri dish with this sample. And because I think my other sample, the really stinky one, has like no chance. It's just gonna turn immediately. But this one, it seems clean enough that I should be able to make like a nice petri dish out of it. And I'm gonna do a floating cover slip in that dish, maybe a couple floating cover slips, and we'll see what kind of interesting biofilms we can grow. It seems like this would be amenable to that. Oh, we got a little Vorticella there. Little dino swimming around. Where'd our little our worm friend go, by the way? Ooh, we got some these. Got some ciliates conjugating there. They always remember if they go tip to tail. If you see them tip to tail, that usually means that they are dividing. And then if you see them mouth to mouth like this, that means they are conjugating. And conjugating is what ciliates do. In lieu of sexual organs, they just swap genetic material through their mouths. This is a nice little scene. Even kind of, yeah. Oh, look. I think these things have cyrtos as well. Cyrti? It's a Greek word. And I think they are like Docked. Cyrtos to cyrtos. These like super haggard ones are weird. Oh, there's that, I don't know if it's the same one, but there's another one of those amoebas. Oh, there's a couple of them right here, actually. There's one. There's one. With the 40x objective. It's cool amoeboid organism here. Yeah, I am, Mike, I am definitely a proponent of fiber internet if you can get it. Oh, this is one of the ones with the spines again. That's so cool. It's got to be related to like Loxophyllum. It just has that look to it. Super cool. Super duper duper cool. Okay, but I kind of wanted to find the our worm friend again. Let's see if we can pull this out. Which one's darker? This one or this one? This one's a little darker. Pop that on. Drop our ISO way down. Even more. And then we will go this way and this way. Shazam. 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 There we go. And brighten that up. Maybe the worm kind of like went off the slide. That happens sometimes. I kind of feel like, oh, I should have ran into it by now. Oh, there it is. There it is. There's our wormy friend. Let's go back to Darkfield. It's neat. Super neat. Definitely, high key, one of the best things about microscopy is just being able to go through things and just change the looks and whatnot. Interesting. Underwater auto. I guess I could use auto. Like auto white balance seems to work a little bit better here. I definitely don't want to use that though when I'm doing my other stuff. Yeah, it's just very blue. Oh, we'll stay like this for a little bit. Oh, hey, Sydney. Welcome. Welcome, mate. Crikey. Have I annoyed you enough yet? That was probably very annoying. I'll stop. I won't do that again. Oh man, I don't know. I just can't like. I like this worm. I don't know how to explain it. It's neat. That's an interesting little like structure there. Ooh, there's a nice pinnularia. Take a little look at the pinnularia here. We can go to. We're not going to get. There we go. Look at that. Look at that. That's a nice little pinnularia frustule. I do believe that's pinnularia. We can go even higher. Now we're in. There we go. Maybe not. Tough to say, honestly. Let's see. We can go polarize, polarize. Shazam. Let's see if we get our. So 400 won't be higher than that, probably. Ooh, interesting. Maybe not pinnularia. It's hard to say. The no, come on now. This button. There we go, and then this. This is how I like it. There we go. Get that nice blue tint. It's got a I forget what they call it, like that central area there. Like it's kind of shaped like pinnularia, but the striae are really, really fine. And it's hard to tell. Hard to tell. But it's certainly pinnularia-ish, and I would bet my bottom dollar it's related to pinnularia. Okay, Amy. Have a good night. Tenetries? Hey, welcome back. That's a name I haven't heard in a long time. Wow. I love a good diatom frustule, let me tell y'all. Yeah, wow. That's really nice, honestly. High key. Nice frustule. A little rotifer there. Oh, there's an interesting little ciliate It's got a little bit of Lacrymaria-ish action going on here. That is... I mean, it's moving exactly like Lacrymaria does. But it's not got the super stretchy neck. At least not so far. But I mean, that is it's got to be it's got to be in the same exact family. It might be a Lacrymaria, but it might not be Lacrymaria lore. It's got the twist in the pellicle, like Lacrymaria has. Yeah. That's an interesting little thing there too. What do we got here? We got an amoeba. A different kind of amoeba than before. This is kind of more of a traditional amoeba, trad amoeba. Ooh, we got some diatoms moving. These are some nice diatoms. I don't actually know what species these are offhand. Maybe some kind of epithemia. Very pretty though. Dang, I would love to watch this thing hunt, but it just kind of seems to be hanging around. Like that. Do a little bit of that. Maximum contrast. Oh, hey, there's our worm again. Just in case you had forgotten how zoomed in we were. Wow, look at those eyes. They're just like little blobules. Kind of looking at the posterior here, looking at those sticky stuff. I'm gonna put another drop of water on the slide. I'm gonna do it way far away from my 40x. There we go. You can see the water rushing in. And this thing kind of sharpened up a little bit. The pointy fella. Something like some colors with it. Whoa, on the move. Look at those cilia go. So I actually on my end, I'm not I don't think there's any actual issue with the outgoing signal, but every now and then when I get like this, the cilia in focus here, like the video stutters a little bit in the OBS presentation. And I think that's it's because it's just like when you have lots of little small details moving, that really takes a lot of extra data. So my encoder's just like struggling. There's its little appendages that it uses to stick onto stuff. Fun to play with these colors. Yeah, Shadow, that's the wrong stream. Kind of a nice. Ooh, that's like super blue. Oh my goodness. Like super duper blue. And this little like organ here, this little like bubble looking organ is also interesting. I've seen similar kinds of stuff on like catanulid worms, but not on like this. I mean, this thing's kind of like what I would call like a planaria-ish worm. Planarid. Wow, look at all these diatoms. Oh, that's an interesting little structure there. I wonder what that is. Wow, look at all these diatoms. And they got these ciliates in here, maybe trying to munch on them. They look a little bit too big. A little bit too big in general. Depth. Go back to awesome. Oh, look at that rotifer. Oh, it's a nice view. All right, add some more water. need to bump the slide, but maybe that'll give this guy a little jolt. Whoa, hello. Dead end. Okay, turn down. Bulldoze over the lesser worm. I suppose if it's like, you know, if it's like nautical rules, you know, then it is the smaller vessels are the ones that have to move out of the way when the big one comes through. So that's on the nematode there. Oh, hey, look at that. That's I think that's the same one. Still over there. A little Lacrymaria kind of dude. Wow, look at this. Look at that cilia. Crazy. Okay. I feel like we spent a good amount of time on this slide. Maybe we could go on to something new. Oh, there's yeah, I don't know if Shahab is still here, but this is purple sulfur bacteria right here. This little globule of critters. That's purple sulfur bacteria. So, there we go. Wow. These diatoms are cool. I like them. Underneath. There's some cyanobacteria. Amoebas. This hair is like interesting. It just kind of looks like an actual like human hair. Maybe it's my hair. Maybe I got hair all over this slide. There's a nice diatom, nice marine diatom with the long apices. Wow, look at that big old nematode. Oh, there's a couple. Well, this one's even bigger. That's too big for the 40x. Let's go to the 20. Quite a scene. All right, well, we're sitting here. I'll make up another slide. I think I'm actually going to I'm going to look through my stereoscope on this one at a petri dish. This is from an old sample from like last week's stream. Get a slide ready here. Enjoy the scene. So I think it was last week I showed a pretty cool, I had a sample with a lot of algae and a lot of ciliates, and that was neat. It's always fun. And I just kind of kept the petri dish around, and I noticed when I looked through the stereoscope today that it's sort of been taken over by one species in particular, and probably not too surprising, it is the common paramecium. And these things are right now, they're so plentiful in this sample that, like, you look through the stereoscope and they're just swarming all around it. And it's like all that's left. And this is kind of a typical end case for a sample I've found, is that, like, often one microbe, and often it is paramecium, kind of take over. Still got plenty of bacterial activity. The diatoms look mostly dead. It's got lots of, like, frustules. Maybe some of these navicula are okay. Well, there's a nice hollowed out skeleton of a copepod. Exoskeleton. Yeah, we got, like, some stray algae here and there. A lot of bacteria hanging around. We're at the 10x objective right now. This is from the Carmel River, right where it comes out and dumps into the Pacific. I'm kind of curious as to where these... often these paramecium will, like, congregate in one particular spot. But anyway, let's just get them, and let's go ahead and we will do polarized light. So the thing is the paramecium just look awesome in polarized light. Just love it. Look how colorful they are. See if you can find a spot where they're all, like, really jamming together. Little bundles of confetti, sparkly confetti, glitter balls. I just love it. I love. You can always count on paramecium for some cool polarized light microscopy imagery. And let's see, we can, we'll bring our polarizer down. Go. And we'll get a wave plate in on the action. There we go. And maybe a little piece of cellophane. It's just the thing. Ooh, ooh. I do like the red. We can get to the red. Come on. Come on, buddy. Very delicate. Very, very delicate. There we go. This isn't quite the same red that I'm used to. Let's see. What if I just turn this? Ooh. It's nice. It's a nice red anyway. Maybe bump this down a little bit. Oops. No. Shucks. There we go. Oops. Oh. One of these days I'll get a proper filter holder for my cellophane. I did have one thing I wanted to try. I have no idea what to expect. This could lead to interesting results, or it could lead to the opposite of interesting results. But we will see. So I'm going to stick a piece of cellophane up under the analyzer. Theoretically, I'm going to do that. Oh, yep, there it is. All right. And then I'm just going to try to turn this cellophane into a orientation. Let me take out the wave plate, actually. I know that. So I just want to, I feel like if I match the cellophane orientations, I should get something. Interesting. That's a different effect. So now I've got basically a polarizer. Ooh. Oh, that's interesting. Oh, that's very interesting, the way that I can kind of control the hue like that. It should be like that. I think, okay, this is definitely worth playing around with. So I've got a linear polarizer over the field lens, and then and then a piece of cellophane, and then I've got another piece of cellophane below the analyzer polarizer. And so this is kind of related to a technique that I was reading about. Just the idea of can I orient these to sort of cancel each other out or do something else kind of interesting. Getting these very pastel colors, which isn't exactly what I expected, but it's cool. I'll experiment like that. That goes crazy. I probably need to really like figure out what I need to make the orientations of the cellophane very specific, so I can experiment with that more in the future. Like, what if I, like, okay, now that's tuned differently. Oh. What do we got now? Ooh, that's nice. That's real nice, actually. I like this because it kind of, it seems to show the color of the crystals really well without them like getting like super bright. Let's see if we can find some more. Where are these guys at? I know they always congregate somewhere. They're always like at the edge of the cover slip or something. I know that I'm missing the big paramecium party. Boom. Well, I get that real super duper orange. Okay, so let me try. I'm going to try turning this some more. I think that should still be over it. Oh, it's kind of off. Sorry, folks. This is the Jason messes around with cellophane for 20 minutes before he goes to bed portion of the stream. Okay. So I'm just trying to like change the orientation to create different effects. I think I'm kind of back. We already had this one. We already saw this one. Maybe did I flip it over? What if I just do that? That'll change something. How did I get that to be like so dark before then? Get in there. Come on now. Oh boy. Okay. Well, anyway, we've been streaming for about 2 hours now. So, I'm feeling like probably a good time to call it a night. Thanks so much, everybody, for showing up, having some fun, enjoying the wonders of the microcosmos and polarized light, which, as I've stated before, is always a clutch technique, always something you should be trying out, and a wonderfully inexpensive technique that can be applied on almost any microscope. So definitely don't miss out on it. Don't sleep on polarized light. All right, everybody. Have a good night. Sydney, E-Packer, Shahab, Amy, Tenatrice, everybody, take care. And